Mic check.
Can you do it once more, because I had... It was okay.
Okay, mic check.
Okay, guys, we are back. Sorry for the technical difficulty. And right now, I've been told that you all can hear Nina, so that's perfect. Let me introduce her once again. Dr. Nina Bert is a researcher at the Duke-NUS Medical University, and she specializes in viral hepatitis with a focus on T-cell immunity or cellular immunity. For her work that we're going to discuss today, we will focus on T cells, but in SARS-CoV-2 infection. And I'm particularly very excited to hear her talk about this. Nina, I'm handing over the stage to you right now.
All right. Thank you. Thanks for the introduction. So yeah, you asked me to talk about our recent paper in JXMed, which is called Highly Functional Virus-Specific Cellular Immune Response in Asymptomatic SARS-CoV-2 Infection. So yeah, the main focus is here on T cells against SARS-CoV-2, but in asymptomatic patients, but we compare them actually to symptomatic patients. Okay, let me just give you a small mini overview of the two arms of adaptive immunity. So we have B cells and T cells, right? So B cells release antibodies binding and neutralizing antibodies, and their main role, or at least of neutralizing antibodies, is that they recognize viral particles directly, and they bind. And when they are neutralizing, they bind them in such a way that the virus can no longer bind to the receptor on the cells. And thus, the B cells and the antibodies that they secrete, their role is actually to inhibit even the virus from entering the cell. So the cells cannot be infected, right? But so we work on T cells, the other arm of adaptive immunity, where we have cytotoxic and helper T cells. It's just slightly different. So they don't recognize directly the virus, but they can recognize virus-infected cells. So once a cell got infected by the virus, on the MHC class 1 complex, so they represent some small parts of the viral antigen on the surface. And the role of the T cells is basically they recognize these small viral particles, so these are small peptides. And then in response, they will secrete cytokines to kill the cell. So they kill the infected cells, or they release cytokines like interferon gamma that help also to present more antigen on the surface, but also helps, for example, to reduce the viral replication. So they act on different levels, but so basically they are different. So the cells still get infected, but the T cell is then eliminating the infection from the body.
Nina, I have a very general question. Actually, I'll let you finish this one.
No, okay. So we have CD4 and CD8 T cells. So they differ in the way they what they recognize. So CD8 T cells recognize the small viral particles, the peptides, the antigen, in combination with MHC class 1. So this is the main difference. So basically you have virus infected cells, so directly the cell that is infected is presenting then this small peptides on MHC class 1 molecules on the surface of the infected cells. So the CD8 T cells recognize these infected cells and is killing them. And the CD4 T cells, which are the helper T cells, they do not directly interact with the infected cell, but we have these antigen presenting cells in the body, right? Dendritic cells, monocyte macrophages, which take up viral antigen that is secreted, and then they process it, and they present, because they're antigen presenting cells, they present this antigen on their surface, but on an MHC class 2 complex, which is then recognized by the CD4 T cells. So this is the main difference, because we have these two, but they are both virus-specific T cells. They recognize directly antigen of the virus. Okay, then I thought to give one small mini slide of background, because even before there was a SARS-CoV-2 outbreak, some people were actually working on coronavirus infections in animal models. So it's not that we don't know anything. So there is especially these two papers that focus on the role of T cells in coronavirus infections. And they showed basically that memory CD4 T cells mediate protective immunity against coronavirus infections. And then there was another paper, earlier paper, that also showed that basically T cell responses are required for the protection from clinical disease for virus and for virus clearance in coronavirus infections. So we know already from previous studies that T cells can play an important role in coronavirus infection, at least in animal models. And then for SARS-CoV-2, I believe sort of direct evidence that T cells can have a protective role comes from this study in rhesus macaques, where they showed, okay, so they show first of all, of course, if you have neutralizing high levels, titer of neutralizing antibodies, this is protecting the animals from reinfection. So this is a kind of convalescent reinfection model. So they have antibodies and T cells. But then if you have some rhesus macaques with low level of antibody, in these animals, they showed basically that then the CD8 T cells are required for protection. So in the case of low antibody titer, when you then have T cells, there's still no reinfection. But if you remove basically the T cells from the monkey, then they can get reinfected and you have viral replication. So I think this is a kind of quite nice study showing that, yeah, T cells can really have a protective role. And then in our lab, I think so we have published 3 papers directly in SARS-CoV-2 infection. Our first paper came out really in June last year, one of the first ones actually studying the role of or the presence of virus-specific T cells in COVID-19 patients. So we showed in this study mostly only that T cells are present, so they get virus-specific T cells get induced by SARS-CoV-2 infection. So we found that especially structural proteins recognizing the structural proteins of the virus are present. We also found that some healthy unexposed donors have some cross-reactive T cells against SARS-CoV-2 already, probably from common cold coronavirus infection, that because some of the peptides are similar, of course, the virus is not completely different, right? Because the T cells only recognize small fractions, small peptide fractions. And then we also showed, which I think was really nice, because here in Singapore, we had access to a group of people who were infected with SARS 17 years ago. And we could actually find that in these SARS patients, you still have T cells that recognize exactly because so SARS-CoV-1 is the most homolog virus to SARS-CoV-2 that ever infected a human, right? So you have actually in SARS-CoV-1 infected previously recovered patients T cells that are recognizing exactly same peptides that are now that are present also in SARS-CoV-2. So we demonstrated that convalescent patients have cross-reactive T cells still 17 years after, again, that could react to SARS-CoV-2 infection. Then we had another paper in Cell Reports, which shows also really that an early induction of SARS-CoV-2 specific T cells is associated in acute patients with a mild disease and a rapid clearance. So I think this is one of the, actually there's very few studies only looking longitudinal in the acute, very early on in acute patients. And we found, yeah, the people who have really mild disease and they are really for a short time only PCR positive, they had actually T cells appearing super early. And then here, this is the paper you asked me to present, where we studied SARS-CoV-2 T cells in asymptomatic patients. So this is actually the vast majority of people. So of course, we speak a lot about the people that SARS-CoV-2 is, I mean, can be potentially deadly, and many people have severe disease, but actually the vast majority of people have asymptomatic infection. And we wanted to understand if they also induce T cells, and if an asymptomatic patients have a proper immune response, that maybe you're also maybe protective for secondary infection. And yeah, this is what I'm going to talk about this paper. So yeah, this is where it was published.
Nina, I just want to mention that I really like the idea that you guys are focusing on asymptomatics, because I think they are the key that shows what exactly you need to do to not get the disease and while being infected.
Exactly. Yeah, exactly. Because it's what we also thought, that, I mean, most people, many studies actually look at the severe patients and so on. Okay, then you understand the drivers of the inflammation and so on. But yeah, at the end of the day, I think what it's as important, or even more important, maybe to understand what immune response you should have to not even know that you got infected, right? So yeah, so we studied the T cells in symptomatic cases, and we compared them with asymptomatic cases. And we asked actually 4 different questions. So first we asked if SARS-CoV-2 specific T cells are also induced in asymptomatic cases. Then we looked at the frequency, if it's similar between symptomatic and asymptomatic cases. We looked if the T cells are targeting the same viral proteins. So because SARS-CoV-2 is a huge virus with many proteins. And then lastly, we ask if they differ in function. And so in Singapore, we had a very peculiar SARS-CoV-2 infection epidemiology, very different to the rest of the world, I would say. Because most of our cases here, so okay, very early on, we had some imported cases, and then some cases in the general population. But the big outbreak was in migrant workers. So here there are many migrant workers, mainly from Bangladesh and India, that work in the construction business. And they live in dormitories, usually 10 people sharing a room, and then the dormitories house whatever 4,000 or 5,000 people. So of course it's a perfect condition for a virus to spread, and this is exactly what happened here. So basically in the dorm, nearly all cases in Singapore were among dormitory residents. And we went then, our collaborators went then to one of these dormitories, and we recruited, I don't know if I put it here, no. We recruited nearly 500 participants. And then we took some blood sample for serum analysis here at the beginning of June, 2 weeks later, and then 6 weeks later. And we just initially studied the serology to see if basically, if more people become antibody positive with signs of infection, because basically in this dormitory, everybody with symptoms was screened for PCR. but people without symptoms, they just were not tested. It was really at the beginning, at the time where we didn't test absolutely everybody. This happened a bit later. That's why now we don't have any cases here anymore. So at this time they tested only by PCR people with any kind of symptoms. But people were really checked in this dormitory, which I think is important, because for this study you need to know, that we are really sure that people have been really asymptomatic, as it's a little bit subjective. But so they were temperature checked twice a day, they had oxygen readings. So I think, you can't, they went to kind of medical checkpoint every day. So I guess, you cannot really go do more than this. And then, so this is a kind of summary of the study that we did. So we invited initially 551 people, almost everybody participated. And then 88% of them completed the follow-up. So probably, among these people who didn't complete, there were maybe some actually even with symptoms that then were moved to other facilities. And on those all of them we did serology. We excluded a few more that had some symptoms that could could come from SARS-CoV-2 infection, even so it's not really sure. And then in a subset, we tested the T cells.
Nina,
So
I have a question
serology.
from the last slide. It's just a general question about patient recruitment. I have not worked with patients yet. So I was wondering, when you send out these recruitment posts, maybe invitations to the people, do they get some kind of monetary reimbursement, or is it completely voluntary with no money involved?
No, in this case here, at the beginning, there was no money involved at all. It was I think at the third time point, we got some agreement from MOH.
Okay.
I think they compensated them with some $10 phone card, something like that. But really something more as a kind of thank you than,
So these people are
I would say.
voluntarily giving some time of their day to get tested and help you study. That's pretty big.
Yeah, because to be honest, there were, at this time there was a lockdown here. Even there was no construction business. There was nothing going on. They were not allowed to leave the dormitory. To be honest, everybody I think was pretty bored in a way. They got anyway compensation for they got paid by the government for the time that they couldn't work. So there was no advantage of being symptomatic or asymptomatic or whatever. From a money point of view, any construction anyway only resumed, I think, in August. And I don't know. I think we have this even among patients in the hospital. I think especially at the beginning of the pandemic, I had the feeling everybody is actually happy if they can sort of do something. I see this even now with the people we study that just get vaccinated. I think people are quite happy to participate more than in other clinical studies.
Yeah, right. No, that's completely understandable. People want to contribute as much as they can in good faith.
I think so. Yeah.
My question, sorry.
Yeah.
My question is, why did you exactly choose week 2 and week the other one, 6 or 8, but not in between or not very early or very late?
There was nothing specific about these weeks, right? It's not that we study. We just take the whole population, these 500 people or 470 something, and we take basically the blood at 3 different time points. It's not that we recruited some where we knew they are infected. We don't know anything. At the beginning, the first time point, we didn't know anything. And then when we did some antibody testing, right, we see here, so we tested with 2 different antibody tests against anti-NP, which is a nucleoprotein. And then we tested for neutralizing antibodies. And we saw, okay, the neutralizing antibodies, we always have a little bit higher percentage. But they're basically at the beginning, 20 to 30% had already an antibody response. So they must have been infected a little bit before we started, probably a couple of weeks before. We don't really know. But not much more before, or maybe 1 month before, but not really much longer, because the first outbreaks here started really in April, and we went the beginning of June. I mean, there cannot be really more than 1-2 months.
So,
And
so
I
did
think
you do any?
it just
So did you do any serological test or RT-PCR to see that if they're infected asymptomatically
Yeah,
before
no,
you
we
treat
didn't
them?
do any Yeah, we didn't do any PCR, so they are not PCR confirmed. We only did antibody tests.
Okay,
So
got
basically,
it.
we assume that everybody who has antibodies got
Mhm.
infected.
Yeah,
This is
that
the
sounds good.
only way we tried, and you
Mhm.
could even see. So basically, after 2 weeks later, even more people were already antibody positive. So there was, and then 6 weeks after was even higher. We were at 50, even with neutralizing antibodies, over 60% of the people in our study became antibody positive. So meaning, yeah, at least 60% of everybody we studied got infected.
Yes.
I think the level is even higher because you can
Mhm.
see that the antibody, I think some that were initially antibody positive then actually also dropped already down. So yeah, there was really ongoing infection in the time where we studied. But yeah, I guess, yeah, why we chose exactly this time point, there's no real, I mean, at this time was more, honestly, more surveillance also, like they wanted to see what's going on. I mean, everybody tried to stop this outbreak, right? So we didn't really, I mean, they tested people with PCR, then they built all these facilities and here at the kind of expo to separate everybody who was positive in the, yeah, they tried to isolate everybody where they really, but only up at this time one was only up on a positive PCR swab. But yeah, these people who are in our cohort, they were not tested by PCR. So therefore
Got it.
not isolated from everybody else.
Okay. Thank you.
Nina, what's the x-axis there? SVNT. What does that mean?
So this is a kind of neutralizing antibody test without using the virus.
Okay.
It was actually developed by one of the groups here at Duke-NUS, but not our team, another team from Lymphavank's team. And it's basically a test that you measure. So you have the this RBD binding domain, right, where on the ACE receptor where SARS-CoV-2 is binding to, and you measure with this test. So you add the serum of the patients, and you see to how much percent the binding of spike is basically blocked to the receptor. So you don't really use. Usually you use for neutralization assays really infect really virus to infect, and then you see how much it's blocked. But then you need to do it in a BSS-3, and it's all complicated. So we use this test that you can do just in a BSL-2 lab.
Okay, thank you.
So basically, but yeah, what it's measuring is basically measuring antibodies against spikes that can inhibit the binding to the receptor. So they should pick up neutralizing antibodies, different type, IgG, IgM, I think it's mixed. And then, so this so this is anti-NP antibody data so I split the patients here over time, and I think it's really quite nice. So you could see the ones that here I have all the ones that were already at the first time point antibody positive, and then you can see that the antibody levels already started to decline. So we could assume, because usually for asymptomatic patients, the difficulty is you don't know when they got infected, right? And I think in this study for the T-cell analysis, we wanted, I mean, what's nice is we could really pick when they got infected, more or less, at least. We can use the antibody profile as a kind of proxy of time when they got infected. So we have the ones that were definitely infected before the study, so probably 1 to 2 months maximum before, because before there was no SARS-CoV-2 in Singapore, so I guess should be like that. Then we got the ones that got positive at week 2, but not here, so they must have been infected within these 2 weeks, or just here. And then we had the ones that were only positive at week 6, so probably they got infected here within these 4 last weeks. So we can divide the study participants in the one that got infected earlier and the one that had a recent infection. And then among those, we picked the ones where we studied the T cells, because the T cells we only studied, because it's much more difficult, okay? So the T cells we studied only at this week 6 time point, right? And then we picked patients, or let's say they are not patients because they are healthy in a way. So we picked study participants that were already antibody positive and stayed antibody positive, here these persistently positives. Then we picked some that just recently converted, so we are sure that they have been infected within the last 4 weeks. And then we used we included some that lost antibody during the study, so probably they have been infected even before the one that were constantly positive. And then we included 13 that never showed any antibody positivity. And then, I just wanted to briefly describe the method with which we study T cells in this paper, mostly actually by ELISpot. So SARS-CoV-2 is huge. It's a huge virus, almost 10,000 amino acids long. And we know already, so when we started this study, we knew already that most T cell responses are actually to the structural protein. So we chose only to study T cells for the structural protein spike, membrane, and nucleoprotein. Where we use peptide pools of 15 amino acid long peptides that cover the entire spike, and the nucleoprotein is a bit bigger, so we divide it into 2 pools. And for spike, because it's so big, we would need to split it into 7 pools, which we didn't do. So we picked the kind of predicted epitopes. So we have a pool of 55 peptides. But anyway, this allows us to look basically at spike, membrane, and nucleoprotein specific T cells by ELISpot. So what's happening is that we add this peptide to your lymphozyme, into your PBMCs on your ELISpot plate, and then the T cells, when they're antigen specific, they will start to secrete interferon gamma that you can pick up by ELISpot assay.
So were you pooling all these, if I understood right, you pulled all the overlapping peptides for a particular portion and then put them in the same well with T cells, right?
Exactly.
Okay,
Exactly.
so you wouldn't know which peptide it's responding to, but you would know it's one of these that it's responding to.
Exactly. Yes.
Okay.
Exactly. I mean, we have then some studies where we go deeper and we try to identify the peptide, but this was not the aim here, because here we wanted to study more kind of,
Oh, I completely understand. That'll be 10 postdocs worth of work. Can I just stop you for a second, because you mentioned interferon gamma was your response variable there. You're looking at interferon gamma secretion. So can we briefly talk about any 3 or 4 major ways how interferon gamma is very important for antiviral responses?
I mean, so interferon gamma is mainly secreted by T cells, CD4 and CD8, I think also by NK cells. So one of their main roles is they activate macrophages. They also activate they upregulate basically MHC class 1 and 2 molecules, maybe more 2. I think also 1. So basically, and there's also some studies that show that interferon gamma can somehow directly inhibit, but I don't exactly know how, inhibit directly viral replication. So it's one of the major cytokines when produced, because we wanted to detect CD4 and CD8 T cells, so it's produced by both of them most of the time. There are some, you can also use other, like TNF alpha or something like that, but interferon gamma is a kind of standard assay to study antigen specific T cells.
Okay.
Good. So what we found then, so we split, as I said, right? We split our participants in the one that was recent infection, persistent infections, the ones that lost antibody. And basically in all of them, what we find, we see T cells against spike, membrane, nucleoprotein 1 and 2, because this one was split in 2 pools, right? So all of them had T cells against all 4 peptide pools. Usually the strongest response we always got from membrane in all of the cohort. And then we found also, even in the one that was persistently antibody positive, these 4 actually are always exactly the 4 same patients. So we are sure that I'm pretty sure they have been also infected, because so they have a T cell response to all structured proteins, or all the 4 parts of the protein that we tested, of the virus that we tested, and to a similar level than the one with antibodies. So yeah, this shows that you can have actually in some people even a T cell response that without actually developing antibodies. So then we compared this asymptomatic people with, so yeah, all our patients here, they must have been infected within the last 3 months. So we infected with we compared it to a cohort of patients that we have here from the hospital that had actually symptoms, most of them still mild, but it's mild to severe. There were, at least all of them were hospitalized. And we see that the pattern is more or less the same. And then it's just to compare for unexposed, where we do see sometimes some cross-reactive T cells. But what is really different is the asymptomatic and the symptomatic, they have T cells to all 4 peptide pools, whereas the unexposed, if they are positive, it's usually only to 1, right? So you can distinguish people who have been infected by SARS-CoV-2 or not, clearly, even if you have some cross-reactivity in unexposed people. But so we see that the strength is similar, but we wanted to see if there's a sort of decline over time post-infection. In symptomatic, we did see that, right? So we studied some during acute infection, and then 1, 2, 3 to 7 months post-infection. I mean, now we have this longer to 1 year, but so basically we see that really at the beginning during the acute phase and here first months where you have probably still some antigen around, the T cell response is super high, it's then decline and it's then going down. But it's still, I mean, if I compare this to other, I usually study on HPV specific T cells, I can tell it's still a kind of really high, I mean, it's a strong T cell response that you can clearly easily test directly ex vivo. And we didn't really see a difference, to be honest, between severe and mild people. I mean, yeah, I think I don't have enough to really split them into groups and so on, and then at the same time points. But I don't really see a difference that severe COVID-19 patients have a different level of T cell response. It seems similar. And then in asymptomatic is exactly the same. So basically, we were able to, right, we have this recently infected, they must have been infected within the last 4 weeks. And again, we see that they have really the highest T cell response. They are probably still acute. I mean, they're probably still positive or just recently cleared the virus. They have the strongest T cell response. And then it's also going down, if you like. These are the ones that were persistently antibody positive, and these ones are the ones that have already lost antibodies. So probably they have been infected a bit earlier than the others. And then also when you compare this side by side, so when we compare the acute to first months, in blue are the asymptomatic and in red are the symptomatic, really there's no difference in the number of T cells. But then it seems to be at 2 to 3 months that the asymptomatic maybe go down a little bit faster, the T cell, total T cell frequency. Even so, I look now at some at 9 months and I'm not too sure anymore. Maybe it's just that they clear the virus faster and therefore initially you have this faster drop of T cell frequency. I'm not convinced, to be honest, that this is really the case at 9 months or 1 year. We will try to study this now. And we can see that actually, yeah, this kind of drop that we here see is actually mainly from the membrane-specific T cells, and the others we didn't. I mean, even so, there's a trend, but there was not really significant difference. So just to sum up this first part, so we find that SARS-CoV-2-specific T cells are induced in asymptomatic cases of SARS-CoV-2 infections, and their frequency is really similar to patients with COVID-19 symptoms. I mean, the other level is the same. They seem to target the same viral proteins, also with membrane always a bit stronger, maybe followed by the NP2 pool than spike. I mean, so there's no difference in which proteins they are targeting of the virus. And the frequency declines over time as it's seen in symptomatic cases. So in terms of the frequency and which there seem to be no difference between symptomatic and asymptomatic cases. And so then we went on to study their function, which I think is, I mean, this is also where we found the difference. And I think, of course, most important part, right? And here we used a different assay. So we didn't use ELISpot, but we used a much, much simpler assay, which I think is really nice in a way. Of course, it gives you less detailed information about which cell is producing what. But so what we do, we use fresh blood, whole blood, no separation of PBMCs or whatsoever. So we take the whole blood and we stimulate directly with the peptide pools. We culture this overnight in the incubator. The plasma, so what we hope or what we think is going to happen, right? The same as in an ELISpot plate with PBMC, so the antigen presenting cells should present these peptides to the antigen specific T cells, and they secrete cytokines, but yeah, then there's even activation of probably the other cells, right? So we don't only study then the antigen specific, the cytokines directly from the antigen specific cells, maybe. But anyway, so we take the plasma the next day, which contains then the cytokines, and we quantified these 10 different cytokines in the supernatants. And then we used UMAP because I think it's a really nice way to see, because we studied lots of samples, right? So we had a lot of patients, and then we studied, right? So many is because we stimulated them with NP, NP2, membrane and spike, and so these are all basically every patient has been stimulated with 4 different pools. So we had actually quite a lot of data.
Nina, in that
And
assay, are there is the blood clotting there, or is it?
No. So we collect the blood in heparin tubes.
Mm-hmm.
So there's a bit of heparin which prevents a clotting. No, there's
So
no
it's
clotting.
just pure blood, no separation. You're looking at serum after that. That's very convenient.
Exactly. Yeah, it's very convenient. You
That's
can
a
study
nice assay.
thousands of patients. No, I do think so.
Yeah.
Of course, and it's fine after quantifying. The expensive part is quantifying then the cytokines, right? Because you need antibodies to, some sort of way to quantify it. But yeah, it's really convenient, and I think it gives actually nicer data. Nicer data than I thought, and really interesting information, I think, because I think one really good part of this is that you don't manipulate anything. You don't need, because many people freeze them down the PBMCs and so on. So you freeze thaw, your activated cells may even die during this process and so on. So here's really, you take the fresh blood, we took it was still warm, if you like. So there's no delay. You really studied that more or less immediately. Yeah. Basically,
to me, the interesting part is that they're all just floating around. All the cells are floating around and presenting and secreting. That there's no need for any structure or adherence. That's pretty cool.
Yeah, no, I think it's nice. So and then basically with this data, so yeah, we did this kind of UMAP analysis, which is clustering then supernatants, if you like, of similar cytokine production together, right? So you can see that. So we studied in total, 274 culture supernatants, and you can see that the ones coming that produce really high interferon gamma and IL-2 are sort of, you find them then here, and the one producing a lot of IL-6 are here. The super high TNF, I1 beta producers are up there, and then on this side here you have the ones that actually produce nothing. And which you can then see when you overlay, where are the unexposed, right? So the unexposed donors are healthy, unexposed people where we are pretty sure they had no contact to any COVID-19 patients. And you can see the supernatants are all, from these people, are all here in this area. Basically, we stimulate with peptides and they don't produce any cytokines. Nothing is happening. Then you have the asymptomatics that are really mostly here on this side. And then we have the asymptomatic, where we have the severe, many of them end up here, these really high producers of IL-6, TNF, IL-1 beta. And then on the other hand, they are more here sort of in the middle, not really on the side. But I will show you something with more, so because we can separate them by time post-infection, right? So in blue, we have the asymptomatic. So we have the one that were just recently seroconverted. So they must have been infected within the last 4 weeks, and they are really here at this side. And if you go back, this is really here, this side where you have this high interferon gamma and IL-2 production. And then, with time, they move a bit more to the left. But in the symptomatic, it's different. You have really in the first months post-infection, they're not here on this side, the high IL-2 gamma producers, right? You have the one with severe disease up here, which are, again, these high IL-6, TNF-alpha, IL-1 beta producers. And then you have them more here in the middle. So they're not this negative, like the uninfected, but they are more here. And then if we compare, then or sort of we put this together, the what we had before, the frequency of T cells that we studied by ELISpot assay with this sort of secretion assay. And because we do think, I mean, most of the interferon gamma and IL-2 at least should come from the antigen-specific T cells. So because T cells are the main producers of gamma and IL-2, we can really nicely see, I think, that here, when we focus on the one that were just recently infected, yeah, okay, they are, as we've seen before, right? The one recently infected, they had the highest number of T cells, but we can also see that they are really the high producers of interferon gamma and IL-2. But for the one that had symptomatic infection, it's a bit different. Yeah, they're still sort of, the production of interferon gamma and IL-2 is not as high. It's different. They're producing less cytokines for sure. And then if I just compare the ones from the first months, right? The first months post-infection. So you have here the orange guys are the one with mild disease, in red the one with severe symptoms, and in blue the asymptomatic. And it's just, again, to compare the interferon gamma level. I mean, especially dramatic, it's between the mild and the asymptomatic, which I think is quite surprising, because you would think a mild disease is not so far away actually from an asymptomatic. But yeah, these people still had fever and cough and a reaction. I mean, they were sick, whereas the asymptomatic didn't know anything. And yeah, you see really a difference in the level. They produce much higher interferon gamma IL-2, but also higher TNF, higher IL-6, higher IL-1 beta. And what I think is interesting also, they produce more IL-10. So maybe they are also more balancing sort of the kind of, that you don't have too much inflammation going on, because for sure they somehow manage to clear the virus without inflammation, right? Otherwise, if you have inflammation, you have fever. So,
it seems like there's a perfect balance there in the middle of severe and mild.
For sure. I mean, there might. So it does seem so. I mean, we see a big difference between the T cells between symptomatic and asymptomatic. So I do think it hints clearly to that T cells do play. I mean, they do seem to play a role in asymptomatic. I mean, in controlling the infection and getting rid of it without causing symptoms.
So we know that the extreme symptoms may be due to the cytokine storm. So do you think that those are not the ones produced by T cells, because the T cells are producing almost the same amount for asymptomatic as well as mild? What do you think about?
Yeah, I mean, I think, okay, when you have severe cases, I think the difference is really, okay, you also have, I mean, for us, it's a bit, the one I studied here, right, they were already convalescent, even the severe in our, so I was actually quite surprised, still find that they produce so much gamma. I mean, that you can still see this sort of cytokine superactivation, even after recovery, right? In a way, quite nice, but you can really see that. I mean, it's IL-6, TNF, IL-1 beta are not really the classical T cell cytokines. It's probably more from neutrophils, monocytes, other cells. And I guess, I mean, of course, if you have inflammation in the lung, in severe disease, you will have T cells also going there. Some will be antigen specific, but you will also have a lot of that are not antigen specific, because this.
So to say that cytokine storm is the reason why COVID-19 patients feel sick, it would be very wrong, because it's a lot nuanced there.
Also, like, I don't really like this term cytokines, because cytokine storm is a bit something different, to be honest. I mean, but no, I think why people feel sick is it's different. It's because you have inflammation in the lung, you have recruitment of neutrophils, monocyte. I mean, you have a lot of things going on. But yeah, clearly, I do think having a functional, at least because these T cells of this asymptomatic seem really functional, they produce a lot of cytokines. I mean, it seems to help, at least, I guess.
Yeah, it reminds me of something that I study autoimmunity. So a common thing in
Yeah.
autoimmunity is that inflammation is good, but chronic inflammation is not, and that's what we
Yeah.
might be seeing here.
Exactly. Yeah. I think so.
Yeah, and so the biggest
Also that
problem with severe cases of SARS-CoV-2 is not actually the virus itself, it's the fact that the lungs are completely destroyed. I mean, the people who are in the hospitals are basically, well, they basically don't have lungs. I mean, even if you give them 100% oxygen, it's just, no use because the lungs are so destroyed. And that might be because of those excess inflammation state.
Yeah. Yes, I agree. Yeah, I mean, it's probably, I mean, it's a kind of mix of also that you don't, I mean, the immune system of people who get even really severe disease means that they don't clear the virus fast, you see. So there's maybe also T cells they recruited, they produce cytokines, but they don't manage to clear the virus. So, and if you have people without any symptoms or mild disease, at least, I mean, yeah, they have an immune response. We clearly see they have also an adaptive immune response. And it's in a way, yeah, these people are faster, also PCR negative, because you clearly get rid of the infection faster. Yeah, I mean, this is only the summary, to be honest, which is our graphical abstract, but I have said everything. So we have the asymptomatic and the symptomatic, and we find that, yeah, the asymptomatic produce less cytokine, which, yeah, I have said, I think, enough. And also this, that the second conclusion is that stimulation of SARS-CoV-2 specific T cells in whole blood of asymptomatic induces this higher secretion of gamma and IL-2, at least, than. And all of this, I do think, hints to that there's a correlation of T cells with protection against SARS-CoV-2. I mean, at least of virus-specific T cells. And it's not because some people thought that asymptomatic individuals mount a weaker T cell response, but this is clearly not the case. The level is similar, and if anything, the T cells themselves at least produce more gamma and IL-2 antiviral cytokines.
All right,
Good.
Nina, that's
So
pretty cool.
these are all the people involved.
Oh, that's a nice lab picture, completely in line with the current meetings, how meetings take place
Exactly.
now.
So which yeah, so basically mostly these are people from our lab, right? So everybody there is here who was involved in the study, but then we have lots of clinical collaborators, mainly from the Singapore General Hospital, NUH and NUS. And then here in this study with the asymptomatic, it was really Liang, Hannah, and Clarence who recruited these asymptomatic patients. And then in the symptomatic, yeah, we have them from NCID and from SGH. But yeah, we have not only these terrible Zoom lab meetings. We had some time in the lab that was nicer, you see. I
Oh,
made another picture for we can have good times here in Singapore. I hope it's
some
coming
people may
back.
say that is photoshopped because meeting people in real is
No,
not possible anymore.
no, it's possible.
Nina, I would like to also draw another parallel with what we are looking at from your study is that symptomatic patients were able to mount a very good immune response early on and probably are killing the virus infection, while the others who are symptomatic, they have an immune response but not enough to clear it, and that's why it's leading to a chronic inflammation. So I would also think it's similar to how anti-tumor immunity works. If you have a strong immune response early on, the tumor is completely removed and you're good to go. But if you have a half-assed immune response, you go through this process called immunoediting, where your immune system selects for these escape variants, and now you are worse off. So of course, there it's not, in our case, immune system is not selecting for viral variants, but it is doing its job incompletely, and that's why you have this ongoing immune response. That's what I would think.
Yes, maybe also selecting for viral variants.
Could also be doing
They
that,
must
yeah.
come somewhere from, there must be a reason why there's some viral variants.
I really like this study. It's because all this time I have been hearing about T cell responses. Yeah, they're there, they're not there, somewhat in the severe, somewhat not in the mild. But your study, I think it
No.
also, because it goes with my bias that there is a good balance required for immune response in order to be successful. So I think maybe that's why I like it more than some others that I have read in the past.
Yeah, so I'm with you.
Okay, with
Yes,
that,
no, I mean, I think it makes perfectly sense. I mean, of course, if you have a, why you shouldn't have a proper immune response if you clear perfectly the virus. It's actually the best immune response you can have, right? The one you don't feel.
right. And our immune system is built in a way that as soon as the antigen disappears, the immune response also disappears. So it's
Exactly.
supposed
So
to
that's
clear
why
by itself.
this big, exactly. I mean, when there was this big drama of, oh my God, the antibody level is going down, of course it's going down. And also, of course, the T cell level is going down because there's no more infection. But therefore, we have memory B cells, we have memory T cells. And I do think, I mean, our first study where we find even, I mean, in the SARS patient now, right? I mean, it's 17 years ago, we still find T cells specific for SARS. And we did some in vitro experiments. So basically, when we take their PBMCs, we stimulate them now with SARS-CoV-2 peptides. I mean, they really proliferate like crazy. So yeah, if they see the antigen again, they will just proliferate and do what's their job to do.
Yeah, related to that, we recently had a blog post in Antibuddies, and the study was that they checked COVID patients after 6 months, and they found that the antibody response was almost none, but there were T cells which were specific to SARS-CoV antigens still present. And one would expect that there will be more of the spike specific T cells, but they found spike specific as well as membrane protein as well as non-structural protein specific T cells too after 6 months, whereas there was no antibody as usual, which was expected. So that was very interesting.
Yeah, even so, I mean, we did study, I mean, we did both basically. We study also antibody here. We do see actually still antibody responses also after. I mean, not in everybody, many people drop. We did one study also in collaboration with another team here, kind of big study. They found that about for neutralizing antibodies, right? They found about that 2/3 lose is also always a bit of a weird term, right? Because I mean, the antibody tests work always like this, that they make a cutoff and they say now it's negative. Okay.
Yeah.
You know, I mean, there's negative and low level, which is completely different, I think. Because you will still have the B cell, also the memory B cell.
Yes.
Yeah.
I think there was, in the US.
And also, memory B cells, they work slightly different from memory T cells, because memory Bs can re-enter germinal center and they can turn into plasma cells later on. And so they may become antibody producing cells afterwards in their second exposure.
I'm convinced that they would.
Yeah. Okay, with that, do you guys, anybody else has any questions? Okay. Nina, thanks a lot for joining us today. That was a pretty good discussion.
Thanks for inviting me.
Yes. So with that,
good
we
that
will.
You guys do this.
Yes, we're doing this twice a month.
Very good.
Yeah, it's my way of reading papers, so I don't slack off.
Good.
Okay, with that, I think it'll be a good point to end the session. Thank you all for joining.
Just a second. Right now we got a question before we end. So John is asking, are your different group age matched? Because most of severe cases from what I know, are old people, just to be sure that what you are seeing is not an effect due to the age.
Yeah, I mean, yes, in a way he's right, of course, and is of course also not perfectly age matched, because of course our asymptomatic cases are younger than the severe, which doesn't mean really that what we find is not true, right? Because it just means that in people with asymptomatic disease, yeah, their T cells are more functional and have this phenotype. And maybe in the ones that get severe disease, maybe it is related with age, that then you don't have this high production of gamma and IL-2. But I can show you, actually, I put one slide here, which is our supplementary, in the supplementary part of the paper, where we split the people by age. Because in our symptomatic cohort, we had actually quite a lot of younger people also. Okay, maybe not among the severe, but among the mild. And when you see here, so basically in the group, okay, these are all time points together, but you see it already here. You don't need to go to the first months of infection. You can see the 20 to 29, we find exactly the same, that again, the asymptomatic produce more gamma and more IL-2. And also in the age group 30 to 39, and also in the age group 40 to 49. And then it's true that, yeah, but for older ones, above 50, yeah, they are not included here because we don't have asymptomatic. The asymptomatic cohort, they were all below 60.
Can I say that it's so cool that you have this data ready?
Yeah, she was so prepared for the question already.
Like that's pretty cool. You're binning them in 10 years of different groups and have that same data show up, the same results, same differences. It's so cool.
It is.
Yeah. No, it is. I mean, I think this is quite amazing, even in the young one. I mean, 20 to 29. Okay, we didn't have a lot. We have only here 4 mild, but yeah, I mean, even here, the 30 to 39, it's about equal. We tested 14 symptomatic and 11 with mild disease.
Okay, Tanu, are there any other questions from the audience?
No, I do not see any.
Okay, then I think now would be a good time to wrap up the session. Thanks everybody for joining. Thanks a lot, Nina, for being here today and walking us through the whole paper, which is very cool. Everybody in the chat, you guys can find the paper link in the description if you want to read it, or this video will be on Facebook and YouTube if you want to watch it again. And with that, I'm going to end the stream. Thank you all. See you 2 weeks later. Bye-bye.